Bachelor of Science Nursing Microbiology, Unit 2, Types of Staining. Microbial identification is essential for diagnosing infections, determining appropriate treatment, and understanding disease progression. Several staining techniques are used to differentiate bacterial and fungal species based on structural and biochemical properties. Gram staining. Principle: gram staining differentiates bacteria into two groups. Gram-positive retain crystal violet and appear purple. Gram-negative lose crystal violet and take up safranin, appearing pink. This differentiation is based on the bacterial cell wall composition, primarily the peptidoglycan layer. Procedure: smear preparation. Spread bacterial culture on a glass slide and heat fix. Primary stain, crystal violet. Apply for 1 minute and rinse with water. Mordant, iodine. Apply for 1 minute to form a complex with crystal violet. Decolorization, alcohol or acetone. Wash for 15 to 30 seconds. Gram-positive bacteria retain the purple color. Gram-negative bacteria lose the stain. Counterstain, safranin. Apply for 1 minute to stain gram-negative bacteria pink. Observation: use a microscope under oil immersion, 100x objective lens. Examples: gram-positive bacteria. Bacteria appear purple color. Staphylococcus aureus, Streptococcus pneumoniae, Bacillus subtilis. Gram-negative bacteria. Bacteria appear pink or orange color. Escherichia coli, Klebsiella pneumoniae, Pseudomonas aeruginosa. Acid-fast bacilli, AFB staining, Ziehl-Neelsen stain. Principle: used to identify Mycobacterium species, e.g. Mycobacterium tuberculosis, which have a high lipid content, mycolic acid, in their cell wall, making them resistant to gram staining. Acid-fast bacteria retain the primary stain despite exposure to acid alcohol. Procedure: smear preparation. Heat fix the sample. Primary stain, carbolfuchsin. Apply and heat over the flame while bathing with stain for 5 minutes. Decolorization, acid alcohol. Wash for 1 minute. Acid-fast bacteria retain the red stain. Non-acid-fast bacteria lose the stain. Counterstain, methylene blue. Apply for 1 minute to stain non-AFB blue. Observation: under oil immersion, 100x objective lens. Examples: acid-fast positive bacteria. Cells appear bright red color against blue background. Mycobacterium tuberculosis, Mycobacterium leprae. Acid-fast negative bacteria. Cells appear blue color. Escherichia coli, Staphylococcus aureus. Special staining techniques: negative, capsular, staining. Principle: used to visualize bacterial capsules, which appear as a clear halo against a dark background. Capsules are composed of polysaccharides and do not retain stains. Procedure: smear preparation. Mix bacteria with India ink or nigrosin on a slide. Air dry, do not heat fix. Counterstain, crystal violet or safranin. Stains bacterial cells. Observation: capsules appear as clear halos around stained bacteria. Examples: Klebsiella pneumonia, Streptococcus pneumoniae, Cryptococcus neoformans, fungus, spore staining, Schaefer-Fulton method. Principle: endospores are highly resistant structures that protect bacteria from harsh conditions. They do not take up normal stains easily. Procedure: primary stain, malachite green. Apply to the smear and heat for 5 minutes. Wash with water. Removes excess stain from vegetative cells. Counterstain, safranin. Stains vegetative cells red. Observation: spores appear green, vegetative cells appear red. Examples: spore-forming bacteria, Bacillus anthracis, Clostridium tetani, Clostridium difficile, lactophenol cotton blue, LPCB mount, fungal staining. Principle: LPCB is used to stain fungal structures for microscopic examination. It highlights hyphae, spores, and reproductive structures of fungi. Procedure: place a drop of LPCB stain on a slide. Spread the fungal specimen with the stain by using dissecting needles. Cover with a cover slip and examine under a microscope. Examples: Aspergillus, Penicillium, Candida albicans, KOH mount, potassium hydroxide preparation. Principle: used to detect fungal elements in skin, hair, and nail samples. KOH dissolves keratinized tissue, making fungal hyphae more visible. Procedure: place the sample on a slide, add 10 to 20% KOH solution, and spread the hyphae with needles. Allow it to sit for 10 to 15 minutes. Observe under a microscope. Examples: dermatophytes, Trichophyton, Microsporum, Epidermophyton. Conclusion: these laboratory methods are crucial in clinical microbiology for diagnosing infections. Gram and AFB staining are fundamental for bacterial identification, while specialized stains like negative, spore, LPCB, and KOH mounts help detect specific structures. Understanding these techniques is essential to have knowledge about patient diagnosis and infection control.